The human body is composed of over 200 cell types, each designated to a particular function. So for example, a pancreatic cell is involved in the function of the pancreas, and the nerve cell is involved in the function of either the brain or spinal cord. A subset of cells, however, are different. These are called stem cells. Stem cells have the capacity not only to make more stem cells, but also to make different cell types. For example, a skin stem cell can make all the cells of the skin. A muscle stem cell can make all the cells of the muscle. And a blood stem cell can make all the cell types of the blood. For many years, adult stem cells have been used therapeutically in the treatment of certain cancers and rare blood diseases. Broadly speaking, there are two types of stem cells: embryonic stem cells that can generate all cell types of the human body, and adult stem cells that have a more limited repertoire. Embryonic stem cells are derived from excess embryos that are left over from assisted fertility treatments, like IVF. The embryos that would otherwise be destined for medical waste are harnessed to study human disease and development in the laboratory. It is important to realize, however, that scientists do not make embryos purely for research purposes. Rather, they use the embryos that would otherwise be destined for medical waste. For example, in the treatment of diabetes or, say, motor neuron disease, the problem is that there doesn't appear to be an adult stem cell that can make insulin-secreting cells of the pancreas, for example, or like motor neurons that are lost in motor neuron disease. Scientists and clinicians, in order to find out what's going wrong and to fix what's going wrong, they need the type of stem cell that can generate the cell type relevant to the disease. So in certain instances, embryonic stem cells are needed. People frequently think about how stem cells can be used directly to treat disease. For example, for many decades, bone marrow and the stem cells they contain, blood stem cells, have been used in the treatment of certain cancers and rare blood diseases. Scientists and clinicians today are even more excited about the indirect use of stem cells as research tools. We can learn more about how stem cells make different cell types. We can use cells derived from stem cells to make better and more effective drugs. So stem cells are, in essence, a source of different cell types that you can use to generate new drugs and learn more about how disease happens. You know, it's frequently said in the media that embryonic stem cells are no longer required because of the advent of a new technology called induced pluripotent transduction. And this is simply not true, because embryonic stem cells will always be needed to study early human development and pathologies and diseases associated with those early stages of development. Back in 2007, scientists discovered that if they introduce certain genes into a cell, they can make a cell forget what it is, so that it reverts back to something like an embryonic stem cell. So it's in a way, it's a little bit like hitting someone in the back of the head and they forget that they're a particular type of person, and they have to relearn what kind of person they are again. But sometimes memories re-emerge, and this is really where the state of the research is now. Can we make a cell completely forget what it was? And can you sustain that amnesia long term within the cell? This new induced pluripotency transduction is a very, very new technology, and we're still trying to find out, how we can make cells forget what they are so that they revert back to this stem cell-like state. But really, it's too early to say for a lot of applications whether embryonic stem cells or induced pluripotent stem cells will be better in future.
Neonatal human dermal fibroblasts and the repro-RNA OKSGM non-integrating vector system. The reprogramming protocol can be broken down into 3 steps: transfection, induction, and selection. The repro-RNA OKSGM self-replicating vector only requires a single transfection. For transfection, we recommend using our repro-RNA transfection reagent. Simply mix the repro-RNA OKSGM vector in a tube with the transfection reagents and incubate for 5 minutes. Take your previously plated somatic cells on a Matrigel or Vitronectin XF coated plate and replace the somatic cell medium with growth medium containing B18R. Add the repro-RNA vector and transfection reagent mix dropwise onto the cells. Gently rock the plate for even distribution. After 1 day, begin the selection process with growth medium containing puromycin and B18R. Change the medium daily for 6 days. At 7 days post-transfection, replace the medium with repro-teaser containing B18R and continue changing the medium daily for the remainder of the induction phase. Over the next several weeks, iPS cell colonies will begin to form. When reprogramming fibroblasts, the cells will change shape, appearing more rounded as the mesenchymal to epithelial transition occurs. Around day 8 to 9, small epithelial-like cells will begin to appear. Around day 14 to 16, these cells will have developed into small clusters of tightly packed pre-iPS cell colonies. And by 3 to 4 weeks, large colonies with ES cell-like morphology will be present. In addition to fully reprogrammed iPS cell colonies, differentiated colonies and partially reprogrammed colonies may be present in your cultures. It is important to accurately distinguish between fully and partially reprogrammed iPS cell colonies to ensure successful selection and clonal expansion of the selected clones. Fully reprogrammed iPS cell colonies generated with repro-RNA and repro-teaser will display the morphological characteristics of embryonic stem cell colonies. That is, they should have distinct borders. Cells should be tightly packed with prominent nucleoli and have a high nuclear to cytoplasmic ratio. To isolate the iPS cell colonies, either a pulled glass pipette or a 22-gauge needle is recommended. Colony isolation should be performed using a microscope in sterile conditions. Sometimes the colonies you want to select may contain differentiated cells or fibroblast overgrowth. These unwanted cell types should be removed from colonies prior to isolation. Drag the needle or pipette around the iPS cell colony to separate the fibroblasts or other unwanted cells from the undifferentiated iPS cells you are selecting. Use of repro-teaser will generate cultures with fewer fibroblasts and differentiated cells, minimizing the need for manual removal of these cell types. To then isolate the selected iPS cell colonies, drag the needle across the colony, up and down, making a grid. Lift and collect the colony fragments with a 200 microliter micropipette and transfer to a new culture dish. The dish should be pre-coated with Matrigel or Vitronectin XF and contain M-teaser or teaser E8. iPS cell lines generated in repro-teaser and then subcultured in M-teaser or teaser E8 typically contain very low levels of differentiated cells as early as passage 2, and are therefore ready for banking and characterization earlier than iPS cell lines generated with other methods. Successful generation of new iPS cell lines should display normal karyotype, express undifferentiated cell markers, and differentiate to all 3 germ layers using either in vitro assays or a teratoma assay.